So today's lecture will be on the chemistry of coral reefs. So this is a new lecture for this year, so I don't know how long it'll take. So if you have to rush out at the end because I've not finished in time, then please do. Or, well, obviously, please stay as long as you can. But, so this will be a bit of a trial, as it were. So we've put in this new lecture on coral reefs because actually, I mean, it was in the news today. It's very topical that climate change and how we're changing the temperature, particularly of the ocean, is really having a detrimental effect on the chemistry, well, on the biology of coral reefs, but actually also the chemistry of reef systems is also really important for this vulnerability to climate change. So these are the things we're going to be talking about today. So mostly talking about the corals of coral reefs, but there are other components too which are important. So just a little bit about coral biology to kind of like to tell you how these guys work. So corals are an animal. They're actually very, very simple animal. They're effectively just kind of a sac with a mouth that is also its anus. And they can feed with these tentacles. They can attract material from the water column and get that in. But they also, shallow water corals also have this symbiosis with algae. Okay, so they incorporate algal cells into their bodies, and those algae can do photosynthesis. So corals can get food from feeding off organic detritus, but can also get food by basically extracting the organic material that these algae are producing by doing photosynthesis within their bodies. Okay? Some corals also precipitate a hard skeleton of calcium carbonate as they grow, so they're basically sitting on this calcium carbonate structure. Not all corals do this, but the corals that tend to form coral reefs do. Okay? There was actually some articles in the news today about this phenomenon called coral bleaching. And what this is when corals get stressed by some environmental conditions, particularly when the water temperature gets very, very hot. Okay? So if there's a particularly kind of warm year, like we've had the last couple of years because of El Nino conditions in the Pacific and Indian Oceans, and this leads to when we have sometimes a low tide, okay, that warm water gets stuck on top of the reef flat and gets heated by the sun even more. And it basically, the corals basically get heat stress and they don't necessarily die, but what they do is they eject their symbiotic algae. Okay, so they lose these symbiotic algae, which are called zooxanthellae, and that leads them to look like they're white, because all of the colour and pigmentation in corals is actually the algae, not the coral itself. So this doesn't kill the coral. Okay, it's still happily sitting there. It's able to filter feed, okay, but it can no longer get any energy from photosynthesis because its algae have been expelled. It's not entirely clear why they expel these algae, but there are kind of a number of hypotheses out there, but it essentially doesn't really matter. What matters is that when the water temperature gets really warm, they can expel these algae. Okay? So it doesn't necessarily kill the coral, but it does make them kind of less happy because they have got effectively less food source because they can't photosynthesize. And actually, if they're out on a coral reef somewhere, there might not be a lot of organic matter in the water column for them to feed on. So they kind of starve to death until the water conditions can return to such that they can reincorporate algae into them and start photosynthesizing again. The corals also become a little bit more susceptible to disease once they've bleached, and that can also lead to them dying, which is obviously bad for the coral. So just a little bit more kind of information on the coral bleaching. So this is this idea that they've got these symbiotic zooxanthellae algae. If you stress out the coral, usually through extreme heat, they eject those algae back into the water column, and leaving the coral just behind us, the animal, without its symbionts, and that basically starves it of food, and it kind of doesn't like that. Okay? So what today's lecture will be about is not necessarily that process of bleaching, but how the chemistry of the water affects the susceptibility of these systems to these extreme events. Okay, so this is an example of what happened the year before last. So we were just starting into an El Nino, and the top map here is not quite a map of sea surface temperature. It's a map of the regions of the Pacific and Indian Oceans that are susceptible to coral bleaching that year. So it's a kind of a combination of the temperature of the water and the degree that temperature is higher than average. So coral bleaching, there's no one temperature threshold because corals that tend to live in warmer environments can survive in warmer temperatures. It's just the added temperature anomaly for that 1 year which increases the risk of these things. And you can see a healthy reef here, okay, nice colourful coral reefs. You can maybe see these kind of little blue tips on the tips of these corals. So Aquapora palmatas. And then there's a bleaching event, and they completely lose their colour. Okay, and actually this reef didn't recover, so you can see the corals actually died, so they weren't able to regain these symbionts. And now you can see that the coral is effectively dead. It's kind of covered in kind of algae. Okay, so you've completely changed the ecosystem through this this coral bleaching event. Excuse me. Right. So just before we go into some of the chemistry, we're just going to have a little bit of an overview of the kind of the broad structure of coral reefs. So we tend to have kind of a coastline. Okay, some coral reefs are on islands which don't have a kind of a central core, but there might be some supply potentially of fresh water and nutrients in river systems to a coral reef. There'll be a lagoon which sits behind the main kind of barrier reef, which kind of sits out to the seaward end. And this is where most of the coral building is going on. And over time, this leads to the front of a coral reef to become quite steep. And this is because basically corals are continually building up towards the surface. That leads to kind of basically this geomorphic feature of this cliff feature. Okay? So things we need to think about in terms of what's affecting the chemistry. So we might have inputs of nutrients and organic carbon from rivers, which kind of come into the lagoon behind the main coral reef. We can actually, just look back, so these kind of regions, you can actually get corals growing in these kind of high sediment environments. Okay, so corals do survive under these conditions, but they're very different systems to the coral reefs that we might see on the outer edge of, say, the Great Barrier Reef or the Belize Barrier Reef or somewhere nice like that. But quite a lot of that sediment that comes down rivers tends to be trapped in the estuary environment, particularly in the tropics in things like mangroves, where all these kind of roots of the mango trees, they've trapped all of the sediment. So some of the factors that control how much of these sediments and how much of the nutrients get out into the ocean is really dependent on some of the dynamics of these systems in estuaries. So if we cut down all our mangrove forests, we might actually increase the nutrient flux out to the reef without actually changing what was going on land at all. So these systems are really important for keeping kind of coral reefs clean. Also, some of the sediment that actually gets out into the lagoon might actually completely bypass the reef, okay, in channels, okay, with higher water flows as they come in and out with tides. So actually quite a lot of the reef kind of areas are really starved of sediment supply. So they're able to be kind of very, very clean, pristine waters because sediment from those rivers might get out into the lagoon, but it might not impact the reef at all because it's just carried straight out to sea in these kind of inter-reef channels. So looking at some of the sediments now, so we're going to have a quick look about all of the kind of things that might be producing sediment and organic carbon actually on the reefs themselves. If we're not getting sediments and organic material coming down the rivers, if that's not actually getting to the reefs, some of we clearly do have sediments. You can see there are sediments in the background of this picture here. So this is being formed actually on the reef itself. We're also forming a lot of organic carbon, so there are things like algae, but actually corals have some organic material in them too. They're made of animals, so those animals are organic carbon. So we're just going to have a look through and see what is kind of forming all of this material. So obviously we have corals, so they're forming organic material, but they're also forming calcium carbonate. And just to recap again, so they're basically this animal sac thingy with these symbiotic zooxanthellae, which is a rubbish Scrabble word. So we also have other things that are producing calcium carbonate. So there are various types of algae, so that are not like seaweed in the kind of like floppy and organic, but actually the algae precipitate calcium carbonate kind of secretion as well. Okay, so these are basically some of these rock algae, so red algae here. Okay. We also get other forms of kind of carbonate-producing algae, so they look like seaweed, and they kind of are a seaweed in terms of their kind of they are algae, but they're producing plates of calcium carbonate underneath a layer of organic material. So these are photosynthesizing, and they're also producing calcium carbonate. There are a whole bunch of other things that produce calcium carbonate as well, so things like sea urchins, sea stars, that kind of thing. They produce calcium carbonate skeletons. Foraminifera, quite abundant on some coral reefs, so these are living kind of stuck on things or in the sediment, and they're producing tests of calcite. Okay, so this is some pink sand from the famous pink sand from Bermuda. If you ever go to Bermuda, some of the beaches are pink, and they're pink because most of the carbonate being produced on Bermuda is actually being produced by Foraminifera living on the sea floor. Fish, bizarrely, also produce a lot of calcium carbonate. So as well as being made of organic material, they process seawater in their gut to basically to moderate the effect of all of the basically the salts inside them. And they precipitate inside them tiny little grains of calcium carbonate. And these are SEM images of this. So when the fish poops that out, it's not only pooping out organic material, but it's also taking alkalinity from the water column and forming this calcium carbonate. So there are various estimates of how important fish are in this process. So this paper came out a couple of years ago now, suggested that it was somewhere between 10 and 90% of all carbonate production in the ocean. Okay, I think they might have improved their estimates since then, but it could be quite a significant source. So we think of like things that produce calcium carbonate being coral reefs, being foraminifera, but actually fish poop might be really significant. So our activities of changing the fish population by eating them all might be having quite a big impact on the carbon cycle that we didn't really think about before. Seagrasses are potentially very important in some regions, particularly because they don't calcify, so they are only photosynthesizing and forming organic matter, and we'll see that that is important later on. So if we just think a little about of the chemistry of these processes that these organisms are doing. So something like a coral is photosynthesizing using its symbiotic algae, and it's also producing calcium carbonate. So when it photosynthesizes, okay, you can see up here, it's taking carbon dioxide and producing organic matter. Okay, so that is taking carbon dioxide out of the water. When that organic matter, when that coral dies or it gets eaten by something, that organic carbon will be released back into the water column as CO2. And in fact, this is a cycle that happens we'll see every day in corals, because the zooxanthellae part of the animal, or the symbiont, is doing photosynthesis, but the coral is an animal, so it's only doing the respiration. So corals, depending on how much of one or the other they're doing, they're either a source or a sink of CO2. When they produce their calcium carbonate skeleton, they're doing this kind of reaction here, which, as you can see, is looks like it's taking carbon out of the system, but it's actually increasing the CO2 concentration of the water. Okay, so production of carbonate is adding CO2, and if we were to dissolve the coral, it would have the opposite effect. It would take CO2 out of the water. So just to have a look about what this does for the pH of the water. So photosynthesis is removing CO2, okay, from the water, and CO2 would have reacted with the water to form carbonic acid, which would then break apart into the bicarbonate ion and release a hydrogen ion. So if we take out CO2, that's going to force all of these reactions over to the left-hand side, and it should, if I've got this right, yeah, it should lower the hydrogen ion concentration, which increases the pH. So the opposite is true of respiration. Respiration, we're adding CO2, so that pushes these reactions over to the right-hand side, and we will be adding hydrogen ions, goes up, and pH would go down. Carbonate production adds hydrogen ions and lowers pH, and yes, and of course, yes, we can fill in all the things as we do. Yes, okay, I guess did you have those arrows in your slide? Yes. Okay, so annotate those arrows on your slide, and that will be an important piece of information you should know. Yeah, cool, that worked. Okay, so let's have a look about how these chemical parameters, pH in particular, are varying on a coral reef. So we're going to go and look at some data from Palmyra Atoll, which is in the middle of the Pacific. It's actually a really weird part of the United States of America. It's kind of an unincorporated territory of the United States. So it's kind of like the president's personal plaything. The Constitution doesn't apply on this island, which is, it's fortunate really because no one lives there, which is quite good. But it's got some kind of like nice, beautiful coral reefs on it. And we're going to look at a study now that basically looked at a couple of sites, one on the lagoon and one on the reef edge. And we can see here that they've measured through time. So along the bottom we've got time in the incorrect American format, so months, day, year. I don't know why they do that. But you can see that the pH, well, first of all, the black horizontal line here and up there, that's the pH of the surrounding ocean, the annual average pH of the surrounding ocean. And the dashed line is the lowest pH in the surrounding ocean for any kind of 1 year. So what we can see here is that, first of all, so the blue line is the pH of each individual sites. So the middle of the lagoon is the top, the bottom is the reef edge, the fringing reef. It's not fringing reef at all, barrier reef. So first of all, we can see that the pH is lower, okay, than the open ocean. Okay, so the blue is kind of lower than the black line. Okay, so this kind of makes sense because we know on the coral reef we're producing lots of calcium carbonate. Okay, that's adding CO2 to the water, and that is acidifying the ocean locally. We can see that actually it's a little bit lower in the middle lagoon than it is on the edge. Okay, so the pH is slightly higher towards the edge, and that kind of makes sense because it's closer to the ocean, it can mix within it, whereas the lagoon is a bit more isolated, so the effects of calcification is a little bit more extreme. The other thing to point out is that there seems to be a lot of variability. Okay, pH goes up, down, up, down, up, down, up, down, up, down, up, down. Yeah? So what's going on here is there's a daily cycle, which is very different from an annual cycle. There's a daily cycle in the chemistry of water on coral reefs. So this is another study which actually wasn't looking at a real coral reef. This was basically a coral reef in a tank. But they exposed their kind of model. It was, like a fish tank coral reef. They exposed it to a day-night cycle of light. Okay, so that's expressed in the blue curve here. So this is kind of the morning. This is nighttime, morning again. And you can see that the blue curve, there's only light during the day. And then the other thing that's plotted on here, so that's the irradiance. The other thing plotted is this G net, which is the calcification rate. Okay, and you can see here that the red and the yellow you can see what's happening here is that during the daytime, okay, the corals are calcifying, okay, and during the nighttime, they're still calcifying, but they're calcifying less. So in the light, corals calcify. In the dark, they calcify but at a lower rate. Okay? So we can look at what else is going on. So that's that same graph up there. So if we look at now, if we look at what happens to, this is the pH. Okay? So in the daytime, the corals are calcifying, but they're also photosynthesizing. Okay? So they're removing, well, it's not just the corals that are photosynthesizing, any photosynthesizing organism, so the algae will be doing this as well. So photosynthesis happens during the daytime. That removes CO2 from the water, and that will raise the pH. Okay, pH will go up in the daytime. But in the nighttime, you don't have that photosynthesis to counteract the carbonate production. So although the carbonate production is less in the nighttime, it still overcomes photosynthesis because there is no photosynthesis. So that causes the pH to fall again. Okay, so this doesn't end up at the same point because of the way the experiment was set up. So you can kind of see the photosynthesis curve in this data, these data, in these data here. So this is the amount of oxygen being produced, and you can see that that matches quite nicely the irradiance. Okay, so when it's light, we get photosynthesis. And that's what's kind of the combination of the photosynthesis and the calcification is what drives this daily cycle in pH. And this is important because the pH of the ocean determines actually how much calcification you can do. So that's what's shown up in this last plot up here. So this is the saturation state of aragonite. So this basically, I think the next slide might show this. Yeah, so the saturation state of aragonite, this term omega, is effectively the concentration of the things that are needed for the formation of calcium carbonate, so calcium ions and the carbonate ion. So the calcium ion concentration won't change because it's a conservative element. It's one component of sea salt. Whereas the carbonate ion will change with pH. So the constant under there is the solubility constant. So it's basically if our saturation factor is greater than 1, we will spontaneously form calcium carbonate. If it's less than 1, it will spontaneously dissolve. Okay, so biology does help it out a little bit, like the formation and the dissolution. But these changes in whether we can form carbonate or not is dependent on the carbonate ion concentration, and that carbonate ion concentration is in turn determined by the pH. So the lower the pH, the less of this species we have, the more of the other ones. At high pH, we increase this concentration. Okay? So why does that calcification matter? Okay? So it matters because if it's harder for the corals to grow their skeletons, okay, if it's harder for them to precipitate that calcium carbonate, it affects their health quite drastically. So this is a study looking at, looking at an experimental plot where they've basically taken some corals and put them in a lab, and then they've exposed them to different amounts of CO2 in the water, which has changed the pH. So we can see here that these on the left-hand side, are high pH. These are low pH. And at the top, okay, well, you can see it. Well, look at the bottom one first. You can see as we increase the acidity, so we lower the pH, the calcification rate, okay, comes down. So the corals are finding it harder to grow those skeletons. They also, because they're struggling with their calcification, they also don't do so well at photosynthesis. They're just less happy in general. Okay? And this is basically adding stress to the coral. So if the top plot is actually the incidence of coral bleaching. Okay? So these corals are becoming increasingly more susceptible to bleaching as the acidity of the ocean is going up. This is another plot to show this. So this isn't corals, but this is a bivalve larvae. So this is something that also needs to grow a calcite little skeleton, shell. It's a shell. They have shells, bivalves. That's not what bivalves do. But you can see here that as they've added different concentrations, 200, 400 parts per million, 800 parts per million, 600, 1600 parts per million, increasing amounts of carbon dioxide to the atmosphere in their experiment. That's dissolved in the water and has lowered the pH, okay? Lowering the saturation state. And as the saturation state lowers, basically these organisms start to have kind of detrimental effects. And the lower the saturation state, the less of these bivalves survive through the larval stage. Okay, so there's lots of parts of the ecosystem are affected by this acidification. So looking at this acidification on the on this kind of daily cycle here, so you might expect that if corals cared a lot about this up and down, okay, this is actually a huge change in acidity. Okay, so that's 0.1 of a pH unit. That's almost that's like a 10% change in the acidity, which is quite a lot. So you would think that actually they should be able to deal with these changes in acidity overall of just changing the average pH by a small amount. But actually, what seems to matter quite a lot is not what the average pH is, but how long, okay, over a long period of time, the pH is above this kind of seasonal average value. So this is what these plots show above here. It's not basically what the pH average is, it's how long, over a long time, the pH rises above this critical value. And you can see that for different types of organisms, it actually affects them in different ways. So organisms that produce calcite and aragonite, so mostly corals, aragonite, calcite might be some of the algae, it doesn't seem to affect them much at all. So it doesn't matter how long you are above this thing, it doesn't really change the calcification rate. But for other organisms, ones that produce magnesium calcite, it can make a really big difference. Okay, so again, the effect is very organism specific. So as we move on and go into the future, as we're ramping up our CO2 into the atmosphere, we are going to change the average amount of the average pH. It's going to carry on going down, but we're also going to change the amount of time that a coral reef is in this kind of stressed state and is able to calcify. Okay, so I guess in summary, we're going to be adding CO2 to the water. That's going to make the water go more acidic, and that's going to add into these processes such as bioerosion. They might change the community structure. We might get different amounts of organic matter, we'll see in a bit. And that's going to lower the pH even further, and that's going to kind of add into the effect of basically degrading the coral parts of coral reefs. So we're going to move on now and talk about the nutrient effects. So these are, coral reefs are typically what we might call oligotrophic, so very nutrient-poor regions out in the ocean somewhere, so a long way from sources of nutrients. So it's a bit odd that they're such fantastic biologically diverse places because there's not a lot of nutrients there for them to survive on. But actually coral reefs have kind of developed through time to actually be very efficient at recycling their nutrients. So actually the zooxanthellae and coral animal symbiosis actually is itself very good at recycling nutrients between the algae and the animal. The nutrients go round and round and round, just like the carbon goes between photosynthesis and respiration between the two. Organisms like sponges play a big role in that nutrient cycling. Watch the video which I posted. It's pretty good. But let's have a think about what actually happens if we increase the nutrient effect. So if we actually start polluting our reefs with maybe some anthropogenic nutrients from maybe tourism or something like that. So we can split the effects into kind of two broad categories. The direct effects of actually what does the coral actually do with these extra nutrients, and also indirect effects in terms of how does the overall community structure change if we add nutrients in, and how does that affect the coral reefs. So broadly, I think the next, so looking at the direct effects, so corals do need some nutrients. So if we have extremely low nutrients, then the corals don't do so well. They need some, but then if you have lots of nutrients, then that seems to affect them in a kind of a bad way. And this is an example of maybe why that might be. So this is some examples of some of the diseases that corals get and that causes them to die. So actually, disease in corals is actually very, very poorly understood. So we don't actually really know what is causing these diseases. We don't even know if it's a virus or a bacteria or even a pathogen of any sort at all. And they have these really kind of like quite obviously descriptive names. So black band disease it has black bands. And there might be different kinds of diseases that just look like that, and we don't really know if they're any different. But yeah, like white plague, white band, and yellow blotch syndrome. Not the most imaginative names of diseases, but they are easy to identify if you give them such broad categories. So what we've got here is an experiment where people went out onto a reef and purposely added some nutrients to different areas of the reef and didn't add them to others. So I guess ignore the external data to start with and think about just the controls and the nutrient additions. So what happened when they added some nutrients? Okay, the prevalence of these diseases went up. Okay, so by adding nutrients, it makes it more susceptible to disease. Okay, it also made a small difference here in the bleaching prevalence. Okay, but this effect didn't kick in until a bit later on. Well, actually, sorry, that's not true. So this is what immediately after doing the experiment, 6 months later, you could still see the effect of this increase in disease. But the good thing is that a year later, this effect of adding the nutrients had gone away. So if we stop adding nutrients to the reef, they can recover. So the bottom is the same experiment, but looking at the effects of how bleaching changed. And this is a little bit more complex in that overall, there doesn't appear to be any change. So adding nutrients didn't change the incidence of bleaching for all of the corals that they studied, but a particular kind of coral. So some corals do show a really clear effect. Okay, so in this Agaricea type of coral, there was a big effect on bleaching from adding nutrients. So again, the mechanisms of this are really poorly understood, but there are clear effects on the actual biology of the coral on adding nutrients. Okay, and one of the things that is thought to control this direct effect is actually the zooxanthellae themselves, so these symbiotic algae. So if we add nutrients, the algae use those nutrients, okay, and either change how much of them are in the coral, so they grow more of them, or they actually change the physiology of the algae themselves, and they might grow slightly differently or use a slightly different ratio of nutrients, and change the chemistry of the pore water, or the fluids inside the coral. But it's really not well understood. So this is a review paper that came out about 10 years ago. And basically, one of the tables in the paper summarizes the effects of adding nutrients on these algae, okay, and on the corals. And I've just highlighted in green where studies show increased nutrients increase the amount of zooxanthellae, and the brown, they say no change. So you can see in the scientific literature, there's no real consensus as to what is actually happening. Similarly with calcification, sometimes adding nutrients lowers calcification. These are all the blue comments. Sometimes there's no effect, and in some cases we see increased calcification. So it's an area of research that basically we don't really understand the mechanisms, but adding nutrients can quite often cause the corals to change in some way, and quite often that change is detrimental. Okay, so we're going to look a little bit now at the indirect effects, which are potentially a little bit more obvious than the direct effects. So basically, if you add nutrients to the ocean, okay, that will promote algal growth because the algae might need some nutrients and they can respond quite quickly. So you might get a lot of seaweed growing on your coral reef if you add nutrients to it. Okay? And if you ever go kind of to snorkeling on a tourist kind of place where there's lots of hotels around the coral reefs, you'll see lots and lots of algae because all of your waste from the hotel probably goes straight out to the sea and fertilizes the algae to grow these lovely algal bloomy things. So the first thing that can happen is that all the algae can be either on the seabed as macroalgae, as like seaweed, but also can be in the water as kind of phytoplankton, and that lowers the light levels. Okay? It's harder for light to get through algal, green, soupy water, and the corals need that light to do their photosynthesis. So that can harm them. The algae on the sea floor can also outcompete the corals for space. So they can effectively just be smothered with algae. So you can see this lovely coral here. So coral with its nice little polyps on it is being smothered by this kind of like blobby green algae here. It can also promote some of the hard algae. So this red stuff here are some of these crustose coralline algae. So these are algae that precipitate calcium carbonate. Sometimes that can be good for coral reefs because that creates a hard substrate which corals can attach onto in their larval stage and then start growing. So the relationships are quite complex, but overall this kind of gloopy green stuff is not so good. And it's not just smothering. There's all kinds of weird, interesting chemistry going on. So seaweed, algae, tend to produce sometimes quite large amounts, some small amounts, different amounts of toxins because they don't like being eaten by fish. Okay, so they produce some toxins to stop them being eaten. And actually, if those seaweeds come into contact with corals, okay, they can actually poison them. Okay, so what we've got here is a study where some people were starting to basically snapped off bits of corals and they kind of grew them in a kind of like a controlled setting where they either left them alone, they strapped some seaweed to them, or they strapped some seaweed extract to them. Okay, and then they looked at kind of what happens to those corals. And you can see here that we've got a control in these experiments for two different types of coral, Porites and Porites cylindrica. They have different ocean basins. And if you've got seaweed, basically at the bottom here we've got the quantum yield, which is basically how well they photosynthesize, and the top is the how often they bleach. So you can see that the control cases are quite good at photosynthesizing and they don't bleach. But if you strap some seaweed onto them, then they tend to bleach more because they've been poisoned and they're less happy. And they tend to be less good at photosynthesizing, so they're dying. Okay? And yeah, similarly for the other species. Yeah. So this is, seaweed is not good for coral reefs. So this is the kind of a potential feedback loop. So we might have some nutrients that start to give us some algae, okay? Start to grow lots of algae. Those algae will potentially poison the corals themselves, but when those algae die, because they've got such a fast turnover of growth and decay, they'll be putting lots of dissolved inorganic carbon, dissolved organic carbon, sorry. So there'll be lots of organic carbon hanging around on the reef now because we're growing all of this lovely seaweed. So that means that some microorganisms will come along and start to eat that organic carbon, and that's just bacteria, viruses, that kind of stuff. They'll say, ooh, carbon, yum, 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 yum. And increasing the basically microorganisms in the water, that can potentially carry coral disease. Okay, so that might kill some more corals. In addition to the smothering and the poisoning, there's maybe biological warfare going on as well between the algae and the corals. Okay? So that might then kill some corals, which might create some more space on the reef for more algae to grow, and it's kind of a vicious circle of coral death, which is super bad. So to summarize these kind of effects now of looking at when we add nutrients to the water, the corals kind of tend to calcify a little bit less. their zooxanthellae density might go up, they might photosynthesize some more, but also these kind of the parts of the life cycle of the coral tend to be kind of hampered somewhat as well. Okay? Similarly, basically just adding sediment to the coral reef has a similar effect to nutrients. It basically is very bad because it basically reduces the light levels, it smothers the corals in sediment and they die. And then the indirect effects as well. So looking at how other organisms and the rest of the ecosystem responds to these additions of something like nutrients. So these crustose coralline algae might do not so well. So that means that there's less places for new corals to kind of like start their little life on. Bioeroders will go up, macroalgae will go up because there's more organic carbon available, so we might get more bioeroding. And potentially diseases going up as well because there's more organic carbon cycling through the system, so more pathogens around. So that's a bit grim, isn't it? So we've got these problems that coral reefs are under a lot of stress, mostly because of rising sea surface temperatures, increasing the prevalence of bleaching, but also some of these other anthropogenic effects. So us adding CO2 to the atmosphere is acidifying the ocean, and that's making the corals even more susceptible to these bleaching events. So these are kind of like the global scale effects. Okay, we're increasing global temperature, we're acidifying the ocean, but also the local conditions are also having a big impact. Okay, so the things that we can change locally, things like adding nutrients. I've not really mentioned overfishing, but overfishing is really, well, fish are really important on coral reefs because they not only have this effect on the carbon system through their carbonate in their guts, but also they eat stuff, right? They eat lots of stuff. So if we have an algal problem, if we had lots of fish, the fish would eat the algae. Okay? So one of the big drivers of change on coral reefs is actually overfishing. Okay, so in the Caribbean, we've removed most of the large fish because they were very tasty. And that has led to changes in the relative proportions of algae versus corals, in addition to us adding lots of nutrients because we've kind of, increased the habitation and the lack of sewage treatment and all that kind of stuff in coastal zones. So what can we do about this? And this is kind of over on this side. So we can kind of look at our how we might respond to these things at a global scale. So the only way of fixing these kind of problems are things like stopping putting CO2 in the atmosphere. Okay, but we can kind of mitigate against some of those things by kind of promoting places in the ocean where some of these corals might be natural refugia, where there's naturally a higher pH, or it's naturally a little bit colder, and those corals might survive. Regions that have got high fluxes of alkalinity, so maybe kind of groundwater seepage coming out of the oceans might counteract some of this acidification. And I mentioned before, seagrasses are important. If we can promote seagrass meadows, that's taking just carbon dioxide out of the ocean, making organic matter, so that's counteracting ocean acidification. So a strategy to get against the ocean acidification impacts on coral reefs might be to promote seagrass meadows, okay, because that mitigates some of the acidification. But some of these local problems, we need to think local about our solutions to that in terms of marine protection areas, so no fishing zones, that kind of thing. But those kind of measures only really work if you can kind of build in some social resilience to the local population. I mean, people will carry on fishing either for food or for an income from tourism. So making the whole kind of interaction between humans and the reefs a sustainable activity is going to be really important. So this is kind of not really what this course is about, but this is kind of like some of the kind of things that you might end up doing kind of with your kind of like geoscience knowledge when you go on to graduate and start kind of like trying to address some of these problems that we're creating with our marine ecosystems. So go and get jobs doing this stuff because it's fun. Unless all the coral reefs die, then it's less fun. Okay, so to summarize briefly the content of this lecture and stuff that you should know. So corals are this symbiotic relationship between the animal and the algae. They can eject the algae when they get stressed out, and that's pretty bad. That's coral bleaching. But the susceptibility, of course, that coral bleaching is dependent on the chemistry of the water, be it the acidity because of carbon dioxide and the nutrient inputs. Okay, so there's more things that you should know over here to do with kind of nutrients. Okay, so yeah, so that's the end of the lecture. If you've not signed in, please sign in. If you have not looked at the discussion forum, look at the discussion forum. It'll provide you with some feedback and to see how you're kind of getting on with the understanding from the lectures. And office hours. Thursdays was the least unpopular choice, so I will be available on Thursdays from I think 12 till 2. If that's totally not working for you, then you can come and try and find me at another time. That'll be fine too. And the deadline for the practical hand-in will be Friday of next week. Okay, even irrespective of which week you did the practical on. So if you've not done the practical yet, come along on Tuesday. It'll be fun-ish. And you can do it then. Okay?